Hello everyone. It is my great pleasure to be here and take part of such an exciting event with so many cool talks and brilliant ideas. Thanks to the organizers for putting all of this together and inviting me. My apologies for not being able to give a live presentation, but as it turns out, unfortunately, I was scheduled to give a lecture at the same time. Anyway, the title of my talk is Markov blankets in the brain, how the situatedness in the environment is reflected in neural patterns. Well, the brain is not like this. It is not in a void, it does not develop and evolve as an encapsulated system or in a vat. It is situated and distributed in a body, which is itself environmentally situated, all part of a multi-scaled system. So when we want to investigate and understand neural activity, it should be with this in mind. My aim here today is to look into how the situatedness in the environment is reflected in neural patterns. The lineup for today is, first, I'm just briefly going to talk about the distinction between two perspectives, neural representation and complex neural activity, and that will set us up to introduce Markov blankets as a tool to look into the different scales of neural activity, such as single neuron, cortical microcircuitry, and networks. In classic computationalism, in cognitive neuroscience, is the view that more than an analogy or a metaphor, the brain is literally a computer and cognition is information-based computations outputting representations. So representations are expected to be found somewhere in the brain. As the authors of this paper tell us, some of the essential features of the cognitive scientific enterprise are, for one, the belief that in talking about human cognitive activities, it is necessary to speak about mental representations. Or two, that the central to any understanding of the human mind is the electronic computer. Or three, the decision to de-emphasize certain factors, such as the influence of affective factors or emotions, or the contribution of historical and cultural factors, and the role of the background context in which particular actions or thoughts occur. While the notion of neural representation is endorsed by many in applied and computational cognitive neuroscience, it is not held across the board. The traditional computational approach is rejected by connectionism, some neurocomputational approaches, ecological psychology, situated robotics, synergetics, and embodied cognitive science or complex and dynamic theory. Tim van Gelder, in his dynamic hypothesis, offers an alternative to the view of the mind as a special kind of computer and cognitive processes as rule-governed manipulations of internal symbolic representations. He tells us that rather than computers, cognitive systems may be dynamical systems, and rather than computation, cognitive processes may be state space evolution within these very different kinds of systems. Walter Freeman and Christine Skarda also note that there's something odd about looking for representations in the brain. They inform us that they've found that thinking of brain function in terms of representation seriously impedes progress toward genuine understanding. Such interpretation is misleading. After years of sifting through our data, we identified the problem. It was the concept of representation. Once we stopped looking at neural patterns as representations of odorants, we began to ask a new set of questions. So in our work, we sort of take on board these insights and call for an eliminativism about neural representations. While the idea of neural representation in applied and computational neuroscience is seductive by giving the impression that it solves a problem, what we realize is that when we refrain from using it, brain activity is not affected. So our aim is to explain neural activity as interactive flows of brain dynamics whose patterns reflect the neural situatedness in the environment. The brain, seen as a complex dynamical system, is then a collection of neural populations that interact with one another in a non-trivial way, such that the collective behavior of the system is different or does not immediately follow from the aggregation of the behavior of the parts. The brain, seen as a complex dynamical system, means that cognitive life is understood as a social cultural engagement of the physiological organism that is situated in the environment in a continuous interaction with the environment. So the complexity of neural activity is not reducible to large quantities of neural connections, nor to the interactions occurring within the nervous system itself. The activity of the population of neurons embodied in brain activity inevitably occurs in the presence of noise or stochastic fluctuations due to thermal energy, ion channel chattering, and irregular synaptic inputs from other neurons. This embodiment also applies to the whole organism, as extended to almost all cells, tissues, and organs of the body, such that, as Denis Noble tells us, organisms also use stochasticity at all levels to generate new DNA sequences, as in the immune system, and new forms of behavior, as in the nervous system. Noise in the brain corresponds to the fluctuations arising from both internal and external sources, for example, of a neuron, cortical column, a region, or the environmentally situated body. Capturing these fluctuations is not trivial, and while mathematics of stochastic differential equations provide guidance in terms of orbits, attractors, bifurcations, a comprehensive formalism that explains the interactive flows at every scale of neural activity in terms of behavior that emerges from the dynamics of the parts of the populations would be epistemically useful. Single-cell spikes are highly nonlinear, but the question is, do such nonlinearities appear in microscopic neural activity, and if so, what processes underlie these nonlinear dynamics across scales? This is relevant because, in the end, what we want to explain is the nonlinear covariance between multiscaled patterns of neural activity and the situatedness in the environment. That means how the situatedness in the environment is reflected in neural patterns. My colleagues and I attempted to answer this in our paper Markov blankets in the brain and the follow-up paper Parcels and Particles Markov blankets in the brain. We applied Markov blankets to the brain to explain the interactive flows at every scale of brain dynamics under a common formalism. We used this statistical tool to explain how embodied systems situated in the environment maintain itself by engaging in dynamics of exchange with their environment. We turn now to the study. A Markov blanket partitions blanket states into active and sensory states. Sensory and active states influence each other directly, such that internal and external states indirectly influence one another. A Markov blanket is not necessarily a physical boundary, such as a cell membrane, but it's rather a statistical one defined by variables that are conditionally independent of each other. So a Markov blanket comprises a conditional dependency, as you can see in equation 1, and dynamics, as you can see in equation 2, by delineating observed behavior as influences between internal and external states. This schematic illustrates the partition of states into internal states in purple and hidden states or external states in orange, that are separated by a Markov blanket comprising sensory states in green and active states in blue. Specifically, it focuses on the dynamic formulation of equation 2. Direct influences are highlighted with dotted connections or connectors, And autonomous states are those states that are not influenced by external states, while particular states constitute a particle, namely autonomous and sensory states, or blanket and internal states. Sensory states, active states, and internal states then comprise the particular states that are constitutive of a functional neural unit. Based upon the neural dynamics used in dynamic causal modeling of canonical microcircuits, we know that synaptic dynamics conform to a Markov blanket, such that internal state corresponds to the conductance of ion channels of one neuron and are distinguishable from the same states of other neurons, but they interact through presynaptic and postsynaptic voltages. This figure illustrates a Markov blanket separating the membrane conductances of a pair of neurons, or between one postsynaptic neuron and all presynaptic neurons. The A terms here are constants that act as connectivity strengths from the active state of one neuron to the external state of another, and from the sensory states of the external state to the internal state of the active state of one neuron. When many neurons are in play, this becomes a connectivity matrix. The sigma function is a sigmoid shape that can be thought of as converting potentials to firing rates. An interesting feature of this structure is that the sensory states, from the perspective of a given neuron, can arise from many different external states, like other neurons, while the active states, which is the membrane depolarization, depend only on the conductance, which is an internal state, of the neuron being depolarized. Normal arrowheads here indicate an excitatory influence, while the round arrowheads show the inhibitory influences. So overall, this means that external states, such as the presynaptic conductance, cause changes in internal states, such as the postsynaptic conductance, via sensory states. This means presynaptic voltage. On the other hand, internal states couple back to the external states through active states, which correspond to the postsynaptic voltage. The Hodgkin-Huxley model's equations describe how action potentials in neurons are initiated and propagated through a set of nonlinear differential equations that approximate the electrical characteristics of excitable neurons in a continuous time dynamical system. Formulating the Hodgkin-Huxley in terms of Markov blankets enables us then to specify how the external state, such as electrophysiological setup, via sensory states, which is the injected current, influence internal states, which correspond to ion channels, themselves then influencing active states corresponding to the membrane potential. In order to keep track with our goal of explaining interactive flows at every scale of brain dynamics under a common formalism that should reflect the situatedness of the organism in the environment, we have to move towards the dynamics exhibited by populations of connected neurons. And this means moving towards a pair of neurons, or a neural network, where current is not coming from the experimental intervention, as we had just now with the Hodgkin-Huxley model, but from another neuron, meaning a membrane potential corresponding to active states of a given neuron, which can be directly influenced by the conductance, meaning the internal states of that neuron. So on top of this scheme is the connectivity of a canonical microcircuit as developed within dynamical causal modeling. And this comprises 4 cell populations with a stereotyped pattern of connectivity. So you can see that from left to right there are ascending connections and the opposite direction descending connections. The dynamics of each neural population, shown here, obey the equations given before, specifying which populations are connected to one another. The second row illuminates how these connections map upon the cortical region. The superficial and deep pyramidal cells play the role of active and sensory states respectively. And in the final row, you can see a separation into network of regions by which the middle two regions act to mediate the far left and right regions. Now, the image in this figure takes the ideas from the previous figure one step further and shows how we could treat the connections between nodes in different networks as dependencies between states in the Markov blanket system. Here, the networks themselves become the active, sensory, internal, and external states. This graphic is loosely structured around the kinds of networks that have been identified using resting state fMRI. Here, we treat the visual networks as internal states that reciprocally influence active states, corresponding to the dorsal and ventral attention networks. The default mode network then plays the role of the sensory states, which then in turn mediate the influence between the above and the external, corresponding to sensory motor network states. This assignment of this is equally valid if reversed, such that you could have sensory motor networks becoming internal states and the visual ones becoming external states. Functional integration has been treated by formal accounts of predictive processing in the brain. This characterization rests upon the asymmetric and directed connectivity that defines cortical and subcortical hierarchies. The study of context-sensitive directed coupling between the nodes of neuronal networks calls for an estimate of affective connectivity under some model of how measured brain signals are generated. One then has to resolve the ill-posed problem of recovering the underlying connectivity parameters of the model, usually using Bayesian inference. And one of the best examples out there is dynamic causal modeling. In this paper, we describe a treatment of effective connectivity that speaks to the emergence of intrinsic brain networks and critical dynamics. It is predicated on the notion of Markov blankets that play a fundamental role in the self-organization of far from equilibrium systems. Using the apparatus of the Renormalization Group, we show that much of the phenomenology found in network neuroscience is actually an emergent property of a particular partition of neuronal states over progressively coarser scales. As such, it offers a way of linking dynamics on directed graphs to the phenomenology of intrinsic brain dynamics. With a particular parcellation scheme based on effective connectivity, we can, in principle, explain and quantify the emergence of large-scale intrinsic brain networks and their characteristic dynamics. But the crucial aspect of the particular parcellization or partition that we employ in this work means that it can be applied recursively in the spirit of the Renormalization Group. So we use the notion of the Renormalization Group to motivate this recursive parcellation because there are some formal constructs in terms of RG scaling that furnish an insight into how dynamics change as we move from one scale to the next. Now, as we have seen previously, the Markov blanket allows one to distinguish a collection of vector states or simply states that belong to something, for example a particle, from states that do not. This provides an operational definition of a particle that, in our present setting, can be regarded as a region of interest or a parcel of brain states. This means that a particular partition becomes a parcellation scheme in terms of functional anatomy. This is where the Renormalization Group comes in a recursive application of the particular partition. In other words, if we start with some states at any level, we can partition these states into a set of particles based upon how the states are coupled to each other. We can then take the principal eigenstates of each particle's blanket states to form new states at the scale above and start again. This recursive application of a grouping or partition operator G followed by a dimension reduction R, leads to the renormalization group based upon two operators R and G. In theoretical physics, the renormalization group refers to a transformation that characterizes a system when measured at different scales. This schematic illustrates a recursive procedure by which successively coarser scale and slower dynamics arise from subordinate levels. At the bottom of the figure, in the lower panel, we start with an ensemble of vector states, here 9. The conditional dependencies among these vector states, the eigenstates, define a particular partition into particles that you can see in the upper panels. Crucially, this partition equips each particle with a bipartition into blanket and internal states, where blanket states comprise active and sensory states, where active are depicted in red and sensory states are in magenta. The behavior of each particle can now be summarized in terms of slow eigenstates or mixtures of its blanket states to produce states at the next level or scale. These constitute an ensemble of vector states, and the process starts again. Formally, one can understand this in terms of coarse-graining the dynamics of a system via two operators. The first uses the particular partition to group subsets of states, while the second uses the eigenstates of the resulting blanket states to reduce the dimensionality. The upper panels illustrate the bipartition of a single particle in the left and an ensemble of particles that is the particular partition per se in the right panel. The insects on top illustrate the implicit self-similarity of particular dependencies pictorially in moving from one scale to the next. Now, this schematic illustrates a partition of eigenstates, which are depicted as the small colored balls, into particles, where each particle has 6 blanket states, which are red and magenta for active and sensory states, respectively, and 3 internal states in cyan. The upper panel summarizes the operators used to create a particular partition. So we start by forming an adjacency matrix that characterizes the coupling between different vector states. This is based upon the Jacobian and implicitly the flow of vector states. The resulting adjacency matrix defines a Markov blanket forming a matrix B, which identifies the children, parents, and parents of the children. Now, the same agency matrix is used to form a graph Laplacian G that is used to define neighboring or coupled internal states. So one first identifies a set of internal states using graph Laplacian. Coupled internal states are then selected from the columns of the graph Laplacian that exceed some threshold. In practice, the examples used specify the number of internal states desired for each level of the hierarchical decomposition. Now, having identified a new set of internal states that are not members of any particle that has been identified so far, its Markov blanket is recovered using the Markov blanket forming matrix. The internal and blanket states then constitute a new particle, which is added to the list of particles identified. This procedure is repeated until all vector states have been accounted for. Usually, towards the end of this procedure, candidate internal states are exhausted because all remaining unassigned vector states belong to the Markov blanket of the particles identified previously. In this instance, the next particle can be an active or sensory state, depending upon whether there is a subset of active states. that is not influenced by another. In the example here, we have already identified 4 particles, and the procedure adds a 5th, which is a top particle to the list of particles, thereby accounting for 9 of the remaining vector states. Now, we think that Markov blanket formalism allows us to explain the interactive flows at every scale of neuronal activity in terms of behavior that emerges from the dynamics of the parts of a population. We thereby think that we can answer how single-cell nonlinearities appear in macroscopic neural activity as nonlinear dynamics across scales. In the end, by virtue of this scale-free formalism, we explain the interactive flows at every scale of the brain dynamics, which then is an explanation of how neural patterns reflect the situatedness in the environment. The formalism presented here can be further applied to the whole organism and even the organism in its environment as covariant fluctuations arising from both internal and external sources. And this work is also consistent with network neuroscience and neural connectivity, as well as dynamical causal modeling, such as models of neural mass models, networks of neural masses, and neural field models. Thanks so much for your attention. I'd like also to thank my excellent collaborators. If you have any ideas, questions, or insights on this, just flick me an email. One last thing, I'd like to take the opportunity to advertise a few special issues that we have open at the moment. For example, we have the Bio AI together that we are putting together with Adam Safran and Andy Clark in Frontiers of Neurorobotics, or the expecting and perceiving how does the Bayesian brain hypothesis stand, which we have open in Consciousness and Cognition with Michael Kirchhoff. And we have 2 coming up exciting special issues. One is going to be edited and published in Neuroscience of Consciousness, together with Mark Miller and Julian Kiverstein, titled Experiencing Well-Being, an Active Inference Approach to Mental Health and Subjective Well-Being. And the other one that is also in the making is with Thomas Vanez, Guilherme Sanchez de Oliveira, and Matthew Colombo. And this one is going to be on scientific realism and modeling in computational cognitive neuroscience. So if you're interested in any of these, do flick me an email. Thank you so much.